Hi, everybody. Last time we talked about the DNA in cells and the two major jobs that have to happen with DNA and therefore the form that they're in. DNA has to be copied and it has to be divvied up evenly to two daughter cells for you to make new cells so that the top of your head to the tip of your toes, every single cell has exactly the same DNA. To copy DNA, cells need the DNA in a form that's like a jumbly mess, chromatin. That's great for copying. It stinks for separating because it's tangled up. So to separate exact copies, they put the DNA into form called chromosomes. Today, we're going to turn our attention to the stages in a cell's life where it is copying and it's in chromatin and it's separating as chromosomes. Now, cells go through 5 phases. There are 5 phases to a cell's life. And different distinct things happen in each of those phases. But they aren't really a real thing in nature. They're just identified stages by scientists. You can think of it this way. You can imagine if I say to you, a newborn, you have certain ideas in your mind, or an infant, right? There's some other images that might come to mind, or a baby, or a toddler, or a kid, or a teenager, or an adult, right? All those things, those different terms, bring to mind certain traits and characteristics that are pretty common to that stage of life. But there's no distinct moment when a kid is a baby and then they're a toddler, or when they're a toddler and then they're a kid, or a kid and then a teen. Like there, as when somebody goes from being a teenager to an adult is not a precise moment. But if I say to you teenager, you have certain ideas about what that means. It's the same thing here. Cells have phases in their lives. It's really just one long part of their lives, but scientists have broken them down into 5 phases. And to remember those 5 phases, I have a little trick. I have no idea why it works, but it's worked for 25 years of teaching. If you use the first letter of each phase in order, it makes it easy to remember. IPMAT. Darned if I know why. It's not a real word, but it really helps. It sticks. IPMAT, IPMAT. So there's 5 phases, and they are interphase, prophase, metaphase, anaphase, and telophase. And you can think of it as IPMAT. Don't write that on an exam for the regents or anything, because nobody will know what you're talking about because it's something I made up, but it'll help you remember the order, IPMAT. So let's talk about what happens here. At first, the DNA is in a jumbly mess. Chromatin, why? Because it's got a copy. You can't put a copy of this chromosome to each daughter cell and this chromosome to each daughter cell and that chromosome to each daughter cell if you don't copy it first. So you have to have copies. So during interphase, the DNA is copied. Now, once it's copied and you want to divvy it up to 2 cells, as chromatin, it's no longer any good because it's too messy. So you have to make it into sticks called chromosomes. That's what happens during prophase. Those sticks are all fine and good for separating. This is identical to that. It's great. This part of the X is identical to that part of the X. Wonderful. But if you start separating them, you are going to accidentally drag maybe this whole chromosome up along with its copies when you split this one. And so the cell must line them up across the middle, and that happens during metaphase. Meta means middle, metaphase. So the chromosomes line up across the middle, and that way when they split, this will go up and that will go down, and we'll have an identical copy go up and an identical copy go down for each of these without accidentally dragging 2 of the same copy in one direction. So they actually split apart during anaphase. And then the cell pinches off into 2 separate cells during telophase. So that's the overview of what happens. I'm going to take you through the phases now in more detail. So we have interphase. This is what interphase cells look like for real. This is an animal cell. And you can see it's got a nucleus and it's got chromatin. You can't really see sticks. It's just a jumbly mess, good for copying. Here's a plant cell. This is the cell wall. And here's the nucleus, and there's the nucleolus. Again, you don't see short sticks. You just have a jumbly mess. It's chromatin. It's good for copying. If you look up here, we have the nucleolus, we have our chromatin, and we have our centrioles. And the cell is going to grow. It's going to copy the DNA, and it's going to copy other organelles as well. That's interphase, I. The next one is P. We're going to go into prophase. Prophase, I have nicknamed the getting ready phase. Now, the DNA is copied, so we are ready to split it, but there's things in the way. If you look, if we have this membrane in the way, we won't be able to split the DNA in two directions. If we have a nucleolus in the way, we won't be able to do it. And at the moment, the DNA is in a jumbly mess, which is impossible to separate it. So the next stage has to get all of that ready. We have to take the chromatin and form it into sticks. We need to get rid of the nuclear membrane and the nucleolus out of the way. And so the getting ready phase, you can see here's a plant cell. This is the cell wall. Here's the nucleus. You can see the sticks are forming. We still have a nucleolus, but the nuclear membrane is starting to disappear. It's a little harder to see here, but here's the cell membrane, and there's what was the nucleus, and it's starting to form sticks. We have our centrioles. They're moving. We have our nuclear membrane disappearing, and we have the chromosomes forming. So we have chromatin becoming chromosomes so that they can get ready. We have the nucleolus and the nuclear membrane, getting rid of them so that they're not in the way anymore. And the centrioles are going to go to the poles. That's so that they can pull apart. They're like fishing reels. They kind of reel in their own copy of the chromosome. So we have a centriole going to one end and a centriole going to the other so that they can pull the identical copies apart when it comes time. So again, this is the getting ready phase. The chromosomes, the chromatin is taken out of that jumbly mess and made into chromosomes, which are easy for moving around. We're getting rid of the junk that's in the way, the nucleus, the nuclear membrane, and the nucleolus, and we're getting the centrioles in place to do the work. That's prophase. So we now have interphase, prophase. So now we're going to move on to M, metaphase. Metaphase is pretty straightforward. The chromosomes line up across the middle. When you look in an actual cell, it's really easy to see. We've got the chromosomes lined up across the middle, the chromosomes lined up across the middle, and that makes it easier to divide them up. So the chromosomes line up. So now we have interphase, prophase, metaphase. We're going to move into anaphase. Ana means back up, to back away. So now we have the chromosomes and their sister chromatids pulling apart from each other. You can see here is anaphase, and these chromosomes are being pulled away from these. Here in a plant cell, we have these chromosomes being pulled away from these. These are all exact copies of these. They were copied in interphase, they were formed into sticks in prophase, they were lined up in metaphase, and now we're getting the exact copies apart so that this end of the cell gets one copy and that end of the cell gets a copy. Same thing over here. These chromosomes are identical to those, and so the two daughter cells will get identical information. So anaphase ensures that both sides get exact copies by separating them and moving them toward the poles. So that's interphase, prophase, metaphase, anaphase, and now we're up to telophase. Telophase is the opposite of prophase. It's putting everything back in order for the cell to go back into interphase. And so we have telophase where the cell membrane is going to pinch in, and we're also going to put the chromosomes back to chromatin. We're going to put the nucleolus back. We're going to put everything back the way it was. So we're going to have a cell membrane form in between, and we're going to go back to chromatin. You can see here in a plant cell, it's going to start to form a new cell wall in between. So the chromatids Go back to chromatin. We're just undoing prophase. The spindle fibers disappear, the nuclear membrane pinches in, or the cell wall forms, and the cell membrane pinches in. So that's telophase. So we have interphase, copies DNA. Prophase puts the DNA into sticks. Metaphase lines them up and organizes them across the middle. Anaphase separates the copies from each other. And telophase returns the whole thing back to the way it was. We'll take a quick look at actual cells dividing by mitosis. These are actual cells dividing. So you can see let's start it again. You can see there's the cell. Let's back up a little bit. You could see the chromosomes lining up across the middle and then being pulled apart really quite fast. There it is. And then being pulled apart. And both daughter cells will be identical because they got exact copies. One more time, you can see the chromosomes forming in prophase, and then you can see them getting lined up and then pulled apart. And that is mitosis.